Assalamu alaikum. Today the topic is the spleen. We will discuss its histology. Firstly, in the spleen, as you know, this is the lymphoid tissue and part of the lymphoid tissue, and it consists of a capsule which is of a dense irregular connective tissue variety. From this capsule, the trabeculae will arise into the parenchyma, or we can say the splenic pulp. And this there are the spaces between the trabeculae, you can see, and these trabeculae will divide the splenic pulp into incomplete compartments. When we cut the spleen in a freshly cut section, we will see it have different colors. For example, red or the whitish gray color when we see from the naked eye. So that is why the splenic pulp is divided into two parts, the red pulp and the white pulp. So we will discuss it under two headings, red pulp and the white pulp. First one is the red pulp. In the red pulp, you can see mainly two structures. Number one are the splenic cords, or you can say the cords of Billroth. These splenic cords have the erythrocytes, lymphocytes, plasma cells, platelets, macrophages, dendrites, and these all cells are supported by the reticular fibers and the reticular cells. Then you will also find in the red pulp a lot of erythrocytes, which will you draw with the red color. Then we have the splenic sinuses. These are the splenic sinuses. As you can see, these were the splenic cords and these are the splenic sinuses. Splenic sinuses, or you can say the splenic sinusoids, these are different from the other sinusoids with some features, because these are lined by the endothelial cells which run parallelly, and between the endothelial cells are the large gaps. The importance of these large gaps is these gaps allow the exchange of formed elements of blood between the sinusoids and the splenic cords. Number 3 is the endothelial cells of the sinusoids. They lie on the incomplete basal lamina. So these are the few features of the splenic sinusoids. So now we will discuss the white pulp. White pulp typically consists of the lymphoid tissue. The lymphoid tissue, it is surrounded by the branches of the splenic artery and forms the periarterial lymphatic sheath. In the periarterial lymphatic sheath, you will find the T lymphocytes, lymphoid nodules, which are mainly composed of the B lymphocytes, and here you will find the secondary lymphoid nodules, mainly with the germinal center. As you can see here, the germinal center. So these are the secondary lymphatic nodule where the branches of the splenic artery is present, and here this artery branch of the splenic artery that run through the periarterial lymphatic sheath is the central artery. You will not draw in the center but on the peripheral side, but its name is the central artery. The white pulp is also surrounded by a marginal zone which have the diffuse lymphatic tissue that have T lymphocytes, B lymphocytes, plasma cells, macrophages, or you can also find here the marginal sinuses. So we discussed the spleen under few headings: capsule, which form the trabeculae, which will divide the splenic tissue into the splenic pulp into the incomplete compartments. Then we discuss the red pulp and the white pulp. In the red pulp, you will find the splenic cords, which are known as the cords of Billroth, and the sinuses. These are the splenic sinuses. And in the white pulp, you will find the typical lymphoid tissue with the secondary lymphatic nodule with the germinal center, periarterial lymphatic sheath, central artery. So this was all about the spleen. Thank you.